to Dan Waite, who is going to introduce today's student invited speaker.
Hey everybody, it brings me great pleasure to introduce Dr. Ann Yoder. I knew I was going to mess that up, and I did, so awesome. Yeah. Well, Ann was chosen by the students to be the student invited speaker because of all the awesome work that she does, and she touches on a little bit of everything: phylogeography, biogeography, speciation. She works with, more recently, hibernation in these lemurs. She kind of does a little bit of everything. It's awesome, it's inspiring, and she's going to tell us a lot about that. So just briefly to give a background about Ann, she did her BA in zoology at University of North Carolina, Chapel Hill, where she first fell in love with lemurs. Then she took a brief spell off to do some work outside of academia and returned and finished her PhD at Duke in 1992. And since then, she's gone to several different places. She's been curator at Yale, she's been a professor at Northwestern, she's done a lot of really cool things. And if you want to know more about her, look at her excellent lab website page. It's very, very well done. It's really informative. But I just want to throw out, we always talk about 2 fun facts for each of these people that we bring in, just to kind of show you something about their personality outside of, you know, the brilliant mind that we get to see speak. And these are both really fun. So the first one was, all throughout college and graduate school, Ann helped with technical theater, and she did lighting for various artists, including Prince, Neil Young, Bono, and Pink Floyd. But at one point during her career, during an Alvin Ailey dance company routine, she accidentally shut the lights off very briefly and heard a loud WTF. For those of you who don't know what that is, look it up later.
But it wasn't the acronym, it was the actual word.
The long form.
Yeah. And then her second fun fact is that, as we'll see, Ann has worked in Madagascar for a very long time, and her Malagasy nickname is Ampela Maheri, which translates to strong woman. And this was received many years ago after a long night of drinking Malagasy rum with the field guides. They were impressed by her stamina. So without further ado, I introduce Ann.
Thank you, Dan, for that. I had forgotten about the fun facts part, email. So I'm gonna try to move quickly through this talk because I always go too long, and I understand that 45 minutes is serious here. But I want to begin by thanking the graduate students so much for inviting me and for Dan for hosting me. It is the greatest honor that a scientist can receive, to be invited by the graduate students, because the graduate students are the ground troops. They're the ones that know all the cool stuff I don't know, and to think that someone as senior career as myself is still cool, it's just awesome. So I'm just thrilled to be here, and I love MBZ, I love Berkeley. I was here 12 years ago, and it feels like not 12 years ago, so it's just great to be back. So with all that, let's just go ahead. First of all, so the title, From Speciation to Sensory Biology to Translational Biomedicine, my journey with the mouse lemur as an emerging model organism. I'm not going to talk about that. I'm going to talk about why I can't stop studying mouse lemurs. Partly this is because there was just no way to fit all these things in to one talk, and especially in 45 minutes without doing short shrift. So I'm going to just I'll tell you what I'm going to do here. So talk outline. So first I'm going to start with a shameless, though relevant, cool story from yesterday and Monday. Then I'm going to talk about how mouse lemurs are an iconic, I left off the mouse part, mouse lemurs are an iconic example of cryptic speciation, and this will lead into some tales of sensory biology. And then a brief history of the taxonomic inflation in the mouse lemur clade, and raising the question of have we gone too far, and so that's sort of where I'm at right now with that part of my science. And then getting to the issue of species delimitation, why does it matter? And I'm going to talk a little, I'm going to touch on some conservation issues, and then I'm going to tell a story about telling big stories with a small sample size of an n equals 3. And it turned into a PNAS paper, so we'll see. We'll see. So we at least pulled it off, and then just ending with building the genomic data resource of my dreams, and that's just going to be a quickie little flourish at the end. Okay, so first the shameless, the relevant story. Okay, so when I was here 12 years ago, I gave a lecture on historical biogeography of Madagascar and how lemurs got to Madagascar, and I hopefully convinced everybody that they got there via overwater dispersal, and I think this is pretty well accepted at this point. Now, how would that have happened, though? This would have been a dispersal from Africa to Madagascar. It's 300 miles of open ocean, so by any measure, that's quite a trip for a terrestrial mammal. How do they eat? How do they drink? How do they manage to survive this? What we see now, currently, these are storm tracks from, I don't know, some authoritative website. So what we see is cyclone storm system where they come from east to west, they sweep across Madagascar, hit the thermal inertia of Africa, and then sweep back west to east, and you can see floating islands of vegetation from these storms. This is what's happening now. It's always dangerous to say, well, it's happening now, so this is what happened in the past. But there was, there's Madagascar, there was a paper published in 2010 in Nature where simulations were done, big computers were used, and the bottom line is that these storm systems were very prevalent at about the time that we estimate that lemurs would have arrived in Madagascar. So it's a mechanism for these overwater dispersals. Now, how would they have survived it, even though it would have been perhaps like 2 to 3 weeks of a trip rather than months? It's still, that starvation time. So it's been proposed that perhaps these were hibernating animals, that they were able to go into a torporous state to survive this trip. So lemurs hibernate. Dwarf lemurs. These are relatives of the mouse lemur, close relatives in the same clade. But this is a dwarf lemur. So hibernation is part of the phenotype of some lemurs, at least, and certainly mouse lemurs. Torpor, they do not hibernate like these animals do. Now, the shameless plug. So, Monday and Tuesday, I was at a workshop called Space Torpor, hosted by NASA, and the whole point of it was to talk about how we can use hibernation biology with the idea of can we induce a hibernation state in humans for deep space travel. I did that for 2 days. It was awesome. And so anybody who wants to hear about it, just it's like pushing a button, blah, blah, blah. It was just an extraordinary couple of days. And Jim heard a lot about it last night at dinner, and he said, "You've blown my mind in the last 5 minutes." So it was just amazing. Okay, so there's the shameless thing over. Now we're going to get into the actual subject. Mouse lemurs as cryptic species. They're sort of classic in that way. So what did we know about mouse lemurs in 1993? There were 2 species of mouse lemurs, one from the east and one from the west, that they were very similar morphologically and ecologically, therefore probably only recently diverged. Both had broad geographic distributions, the whole west coast of Madagascar, the whole east coast of Madagascar. Both readily adapt to secondary habitat, and it's true that you can find them in virtually any shrubby little forest or woods outside of a village. There will be mouse lemurs there. And so all of these things indicated low risk of extinction. So that was our understanding of things in 1993. Well, fast forward to 2016 and now 2018, we have over 25 named species of mouse lemur. So we've gone from 2 to more than 25 in the space of, a couple of decades, not even a couple of decades. So what's up with that? Well, for one thing, there's pushback from the biological community because they are morphologically, at least to our eyes, pretty much indistinguishable. They're just little brown jobs, and they'll fit right in the palm of your hand. They're brown, they're nocturnal, their behavior is very similar, their ecology is very similar, and they're all over Madagascar. And so one can easily imagine that they're just one big metapopulation. So why are we calling them species, especially when we can't tell them apart? Well, one thing I always say is it doesn't matter if we can't tell them apart. The question is, can they tell each other apart and choose the right mates and so on? So that's really the biology underlying this. And I just like to use this illustration. It's from a paper published by Barley et al. In Evolution in 2013, where they're talking about cryptic species and species delimitation. And that, on the x-axis we have genetic divergence, on the y-axis we have morphological divergence. And so there are two areas where delimiting species is very difficult. There's this sort of, line in the middle where it's relatively easy, but it becomes difficult if you have high levels of morphological divergence but very low levels of genetic divergence. Sort of cichlids would be sort of a classic example there. Or on the alternative is where you have low levels of morphological divergence but very high levels of genetic divergence. And so that's where the mouse lemurs reside. And so my work is validated in that it's problematic but conceptually interesting. So here we go. So, just again to make the point, seeing is not always believing, and mouse lemurs are nocturnal. So I would posit then that sight, which is we're very visually oriented primates, so I think we're predisposed to expect to see differences just because of our innate expectation of our conspecifics. But, let's just imagine you're a mouse lemur. It's probably a different kind of sensory world if you're nocturnal. So that means that sound might be quite important, acoustic communication. And this is not my own work. This is work from Elke Zimmerman. So this is a gray mouse lemur, Microcebus murinus. And this is the mitochondrial gene tree. So this is just their phylogenetic map. So here's the male advertisement call from this gray mouse lemur. Contrast that with the Goodman's mouse lemur. I always play that one twice. Quite a different sound, I think you would agree. And here's yet another one. I call this one the construction worker. Not only have they made, I love that. Not only have they made these recordings, and these are ultrasonic sounds, so they have to slow the tape down so that we can actually hear it, because it's meant to avoid predation. So they're communicating at a sound frequency that is invisible or not audibly perceptible to us and to their predators, one hopes. But also, so Elke and her colleagues, in addition to just doing these recordings, they're doing sonographic analysis. And just to show you, that was mouse lemur 1 and mouse lemur 2, so you can see that quantitatively these are very, very different calls. So this, I think, speaks to mate recognition and prezygotic reproductive isolation. So this is putatively a mechanism whereby they're maintaining these species boundaries. So, and I thank Elke for those calls. So moving on to smell. So I got interested in olfaction as another, so Elke was already, taking care of the sort of acoustic world. I thought, well, let's, look into the olfactory world. And mouse lemurs, as is true of all strepsirrhine primates, which are the tooth-comb primates, the lemurs and the lorises, have a wet nose, like your dog or your cat. So olfaction is, just kind of readily obvious that, olfaction is more important to these organisms than they are to ourselves with a dry nose like the amphipoid primates, haplorhines. So we got into the Bromare nasal receptor subfamily of pheromone receptors. So I'm just going to call them V1Rs because that's a lot easier to say than Bromare nasal receptor class 1 over and over again. So what are Bromare nasal receptor genes? They are G protein coupled receptors. They are found in this little structure called the vomeronasal organ, which is above the palate in almost all vertebrates. There is at least a trace of these cells, and it's quite developed in lemurs, and all lemurs, that this organ is there. And there's a straight one-to-one connection, so the receptors are here, and then there's an immediate connection to the anterior olfactory bulb of the brain, and that mediates behavior. So there's big literature about these V1Rs being important to mediating species boundaries and perhaps driving speciation if it's reinforced. So we got interested in that. And there's a growing body of knowledge about the specific gene functions of the particular Bromare nasal, the V1R alleles, but in mice. So what have we done to try to build this into the mouse lemur system? So we did a sort of a shotgun approach to this. We just designed some primers. We pushed Sanger sequencing to its very limits. This was right on the edge of like the transition into next-gen methods, and we chose to do targeted PCR sequencing and Sanger sequencing. So what we found, though, was this previously undiscovered radiation of these V1R genes that were specific to the lemurs and the lorises. They're not found in any other primate or any other mammal. So all of these black, and these were picked up. These are obviously not in this clade, but these are all sorts of other mammals, primates, rodents, bats, you name it. So there's this huge proliferation of these gene copies that are specific to the strepsirrhine primates. We named it the V1R strep. I've never named a species, but I have named a gene subfamily. I think we're the only people that call it V1R strep, but maybe you will too now. So what about this? So first of all, this is cool. It's obvious that olfaction is an important, or chemo sensation is very important to these organisms, it seems. But the question that drove us to this was, is there a difference between V1R abundance in nocturnal species like mouse lemurs versus the diurnal lemurs, which presumably are more visually communicating, and indeed they have all kinds of coloration, pelage variation, and so on. So what did we find? When did the expansion happen? Well, it looks like in the mouse lemurs. So if you look at the intact copies, it's up in the range of about 100 in the mouse lemurs, and it's also quite high in the dwarf lemurs, but if you look at these diurnal lemurs, it's much lower. The eye is odd. This is a nocturnal primate, very low. And also, just as a caveat, it looks like the sister lineage to the mouse lemurs, Mirza, which is not speciose, but is also nocturnal, also has this expansion. So we're working on that as we speak. My technician, Kelsey Honeycutt, whose name is cut off down here, is working on this as we speak. And she's been doing a lot of mapping to our genomes. So what has she found? She has found, by mapping, so this is the phylogeny of all the V1Rs that she's mined out of the genomes. She has found that 57 of 102 genes, so 56% of these V1R loci are found on this one tiny chromosome, chromosome 22. I don't know what that means. We're going to look at that, but it is quite an interesting finding. And this is the first time that these alleles have been mapped to the chromosome structure of the genome. So these are novel data. Okay, so the phylogeny of these alleles is quite informative. And I'm going to walk you through that. So this is work from Catherine Dulack's lab at Harvard, and they do a lot of transgenic mouse work. So they have actually done the assays to expose mice to different odorants and pheromones, and then they kill the poor creatures, and then they're able to map exactly which alleles are being upregulated by the different stimuli. So what have they found? So here is the phylogeny of the V1Rs in the mouse, and they have found that in this clade, right here, the V1R subfamily is receptive only to the stimuli of female mouse bedding. I mean, that is hyper-specific. It's not male, it's not any other organism, it is female mouse. That's it. That is the only thing that will trigger a response in that particular locus. However, in this clade, this subfamily responds to female mouse bedding, snake bedding, and ferret bedding. So here is a subfamily or a clade within the subfamily that is very open to, it's receiving information about predators as well as conspecifics, so it is doing something quite different. I would love to know this kind of information for mouse lemurs, but we can't do the kinds of destructive experiments that you can do in mice and mouse lemurs. So what are we doing? Well, we are using a multi-reporter back transgenic mouse line to express V1R strep genes. And just the basics of it, and we're just getting going on this. So we identify the V1R strep in a back library, courtesy of Peter DeJong, who has the backpack resource nearby. So we identify those, we cut and paste the sequences into transgenic mice, and then we propagate the mouse lines, to then do downstream analyses to look at function. So at this point we have 3 lines going, and they are, so we have the 3 lines going. Here's the good news. It has been tested in all 3 lines that gene expression is occurring. So that's just a baseline. So we have managed to get all of the works from the back library into the mouse genome. So we've got the regulatory elements, et cetera. That was the big question, one of the big questions going in. Would we be able to do that? And we have been able to do that. That's where the project rests right now. So this work is being done in collaboration with Hiro Matsunami, who is at Duke. He discovered the V2Rs. He is just a genius. I love working with him. His graduate student, Kevin Zhu, is the one who's actually doing this as a side project. And so what we want to do ultimately is examine whether lemur-derived stimuli can influence the behavior of these transgenic mice that have the V1R streps in there. And so we can ultimately, get at some of these questions of, specific function. And so I asked Hiro, I said, what can we hope to find in the system? And he said, well, this is probably too much to ask, but it would be super exciting if, for example, the transgenic mouse started attacking or mating with a castrated mouse painted with lemur soil bedding. So that's the end goal. What we're first going to do is we've collected a bunch of stinky stuff, and I think this is going on right now. Kevin is working on this, where we're just going to present stinky stuff to the mice, and then measure, just do RNA-seq. And just see if gene regulation is being affected. And so that'll be step 1, and that's ongoing, and I can't wait to know the answer. So moving on. How did it all begin? This is going back in time. It was about 1998, and I was on faculty at Northwestern, and collaborating quite a bit with Steve Goodman, who is a field biologist in Madagascar, who works for the Field Museum. And I got an email from Steve, and he said, I'm working with a Malagasy graduate student who is really bright. He's doing morphometrics and field ecology, and he is certain that he's seeing multiple species in western Madagascar. And so we have voucher specimens, et cetera, and we want you to do the genetics to prove it. And at the time, and I still am traumatized by species concepts, and I had a really nice conversation with Dan about said trauma. Brent Mishler, I don't know if he's in the room, but he's responsible. Going back to root causes. So anyway, so I was like really intimidated by this whole idea, but I could not resist the idea of examining, genetically more than 100 wild-caught samples from Madagascar. So I said yes, and off we went. And I won't belabor it, but I'm just going to jump to the point. So we came out with a couple of nice papers. I led the genetics paper, and Rodin Rasula Arasan, who was the Malagasy graduate student, led the morphological species descriptions, and we published the papers sort of side by side. So we identified all these species. I was shocked, at the amount of genetic diversity that we found. But there were also some interesting phylogeographic results that surprised me, and namely, this is the point being, so this was a PNAS paper that was based on 2,400 base pairs of mitochondrial DNA, and this was in 2000. So we've come a long way. Everybody now is like, we have 500,000 SNPs. It's like, right. Well, and I do too now. So it's a different world. But so what was really surprising to me was, rather than having an east-west split, which was the traditional taxonomy, it looked like a north-south split, and that was just very puzzling to me, and led me to all sorts of things. And I'll just tell you that when I was first doing the first phylogenetic analysis, I was trying to root. So we had largely looked at samples from the west, and I was trying to root the tree with some rufus from the east, and you could not root the tree in such a way that rufus came out as a clade. It just was impossible. So I finally, gave up and said, well, okay, there is no east-west separation. It's more of a north-south thing. So that was a big surprise. And so I present this sentence from the paper for a couple of reasons, and I'll explain. So this result casts doubt on traditional ecogeographic models, whereas also suggesting that species diversity may be underestimated for eastern as well as for western mouse lemurs. And so there are 2 points here. I use the words ecogeographic model. I see myself cited as Yoder's ecogeographic model with some frequency, and there is no model. It's like there are no parameters to this model. It was just language. And the other consequence was that there was suddenly a stampede of investigators in eastern Madagascar collecting mouse lemurs and looking for species diversity in the east. So my point to the graduate students is, be careful what you say in published form, because actually people read it and stuff can happen as a consequence. But anyway, yeah. So yeah, remember for later the surprising bit about the east and the west. Okay. Next steps, of course you want to know the age of things. I always, a time tree is like the essential piece of information you need before you can ask any biological question in my worldview. So the next thing I did was I collaborated with Zihang Yang, and we published this paper in Systematic Biology entitled Comparison of Likelihood and Bayesian Methods for Estimating Divergence Times Using Multiple Gene Loci and Calibration Points with Application to Irradiation of Cute-Looking Mouse Lemur Species. That was in 2003. Zihang is the one he's responsible for the cute-looking mouse lemur part of this title. Anyway, so we had an email discussion about whether we should, actually use this title, and so we decided, oh, well, let's try. And so we had, an AE, we had 3 reviews, lots of feedback and everything, and we're looking for someone to say that the title was ridiculous and we needed to change it. There was one reviewer said, the title seems a bit long. So we just chose to, forge ahead. But the biological point is that we estimated the mouse lemur radiation to be approximately 9 million years old. And so that was, for me, a big surprise because, again, this was still in the old thinking that this was just, these were very closely related, recently diverged organisms. So this changed my thinking in various ways. Next step, and fast forward to 2010, work led by Dave Weisrock, who's now at University of Kentucky, was a postdoc in my lab. We expanded our data set to many individuals, 284 individuals, almost 80 localities. 23 were new to the study. We went beyond mitochondrial loci to include nuclear loci, etc. And what did we find? So one thing that we found was, whereas with the mitochondrial DNA, you get these very nice clades. So the coalescent times are perfectly in alignment with these species boundaries. With the nuclear loci, that wasn't the case. You would sometimes see the same resolution with the nuclear loci as with the mitochondrial, but not consistently. And so there were some consistent patterns, but there was a lot of inconsistency. So we knew that we couldn't use this argument of monophyly with the nuclear loci to support our species identifications. So this was early in the days when people were using structure as a tool, and Dave thought that would be a nice way to look at the nuclear signal and its congruence with the mitochondrial signal. So what you see here is the structure plot mapped against the mitochondrial tree. So what we found was that there was really nice congruence with these mitochondrial clades and with these structure plot allele frequency groupings. So it seemed to support our hypotheses. You'll also note, so this is the gray mouse lemur here. You see quite a bit of diversity. And I also want to point out, so when you're looking at a structure plot, and I'm not sure that everybody keeps this in mind, it really depends on which loci you're looking at. If you're looking at rapidly evolving loci, you're going to see structure a lot faster than you will see with slowly evolving loci. So we're looking at like nuclear exons here and a couple of introns. So the fact that you see this much structure in these nuclear data is actually a pretty big deal. So this is deep. So what does that mean here? It was just something that we puzzled over. But one of the points was that this was primarily a validation exercise. This was not a species description. It was just, let's look to see if the nuclear loci support our mitochondrial hypotheses. Okay, so fast forward a little bit further. We, in 2016, actually described 3 new species, and I was next in author because this is Scott Hotaling is a student of Dave Weisrock's, and so Dave was the senior author on this, and I was just along for the ride. So we used all the same genetic data as previously, but we used BPP, the Markov chain Monte Carlo approach developed by C.K. Yang and Bruce Ranalla, and base factor delimitation tests. So this was entirely a genetic species description. And this is, these are sort of early days for these kinds of things. And there's a lot of debate in the community right now about what, is this a sound practice or not? We got publicity about this. It was written up in all sorts of places. So we were kind of like, well, people are noticing. So, again, just re-emphasizing that the species diversity has really exploded. And I'm wondering, is it time to walk it back a bit? And I'm going to tell you why I'm asking that question right now. So just, this is the mitochondrial tree from the Hotaling et al. Paper. I just want to point out where the new species were described. And it was this same place where we had seen this deep structure and nuclear loci in the previous work. So that's where the new species came from. And I want to point out, so this is, like if you go on the IUCN website and you look for the distribution of Microcebus murinus, the gray mouse lemur, this is what you will find. But the updated taxonomy will show you that Gans horni and Manitatra, which were the 2 of these new species, are here. And so you can see that there's a discordance or disjunction in the distribution, and it's historical. It's not human mediated. So there has been separation of these populations for a long time. So I thought, that would be good validation for our species descriptions. However, I was on a conference call with some of my German colleagues, including Elke Zimmermann, and we were talking about this species, in particular Gans horni, and they had an image. I said, this is a simulation of unpublished data from Zimmermann. This is actually taken from an ancient paper of hers. It's just a sonograph. But what they showed was that the male advertisement call actually is identical for Murinus in these two, putative new species. So I thought, uh-oh, maybe we need to step back and take a closer look at this. So we're in the process of doing that. And I just want to emphasize that this is really quite relevant right now. There's just a lot of discussion in the literature about species descriptions based entirely on genetic data. And this paper, of course, I'm sure almost everyone in the room is familiar with the Zuckerman and Knowles paper. And I saw this multi-species coalescence delimit structure, not species. I was like, oh God, please don't cite our paper in there, and they didn't. So I'm sure they were just being nice. But anyway, so it's really worth thinking very carefully about this, if this is a good thing to do. And just so you know, vouchers, I'm a museum rat, totally. I'm all about voucher specimens. You can't voucher lemurs anymore. It's just not possible. But we do have vouchered DNA for these new species at the Duke Lemur Center, so there's something. Okay, why does this matter? So let's get into conservation just for a second. Madagascar, I'm sure you know, is the poster child for rapid deforestation and human-mediated landscape change. The royal we at the Duke Lemur Center have a big conservation program in the northeast parts of Madagascar, the Sava. And in the Sava, so this is the Sava, so this is basically the Sava here. So there are two protected areas, Marjeji and Anjaraharbe Sud. I can hardly ever say that word. So these are actually protected areas, but you can see there's this corridor connecting them called the Kamatsa. This is not protected. So we're very interested to know how is this corridor working? Is this a critical means for dispersal and sharing genes and continuity of species, etc. So that's an active program that we're working on. We're doing biodiversity inventories in there. But I thought, well, this is a great opportunity to collect mouse lemurs samples. As that's always my default setting. It's like, oh, I can get some mouse lemur samples. So that's the question. So enter Marina Blanco here, who is our on-the-ground person in Madagascar for the Duke, on the salary of the Duke Lemur Center. She is a field biologist extraordinaire. So she's leading this biodiversity survey in the Kamatsa, and I said, collect every mouse lemur you can while you're there. And so she did, and here's they sort of freeze when you grab them. It's like a cat, there they are. They don't feel it. So she went, this is Marina's slide. So she went to 5 field sites, collected 29 mouse lemurs, 7 dwarf lemurs, took 4 flights, and traveled 4,200 kilometers by road over the course of our summer in 3 months. And I just, and you're thinking, 29 mouse lemurs, 7 dwarf lemurs, that's not much for all that effort. Well, here's what, travel in Madagascar looks like. It's just not easy to get anywhere. So, my hat is off to Marina for doing what she was able to do. So I got an email from Marina at the end of, she was about ready to come back to the U.S. and she said, she's sort of reporting what she's been up to and, her plans and so on. And there was this little phrase, then, depending on the return date to the USA, I will try to fit another short expedition into Ankapu Bay to try to get mouse and dwarf lemurs. And then she explained that it was just an hour's drive north of Tana, which is where you have to fly out of. And, I was like, oh, sure, go for it. Why not? It's a day trip. But let me show you what Ankapu Bay looks like. And this is why I was dubious that she would find anything. There's Ankapu Bay. We're going to pull back out, and this is the landscape, so it's like really seriously lemurs in that kind of place? And also, it's tiny, it's 1 kilometer in length. It's just this tiny little fragment. And it's curated by the Missouri Botanical Garden, by the way, which is quite interesting. They're doing very good work. Well, fast forward, we get the samples back. She collected 3 mouse lemurs from this site, from Untitled Bay. We just did a quick, and when I say we, it's Peter Larson did this. And what we discovered was the genetic diversity at the mitochondrial locus was the highest in this tiny little forest fragment than any other site that had been sampled for this particular species of mouse lemur. So that was just like crazy. And that's when I stopped and went, whoa, this is really interesting. So previously, this species of mouse lemur, Lehi-Lahatsara, which means Goodman's mouse lemur, is named after Steve Goodman, it was thought to be a rainforest edge specialist because all of the collecting sites had been along the eastern escarpment, right up against the rainforest there. But suddenly, we had Lehi-Lahatsara from central Madagascar. So this quickly revised our understanding of Lehi-Lahatsara's sort of natural condition, and we went from this sort of edge specialist to the idea of a high plateau specialist. And just to give credit where it is due, Uta Radespiel, who's one of my colleagues in Germany, had also suggested some time ago that Lehi-Lahatsara might be a plateau specialist because she had seen other symptoms of that. So, you know, this is convergence of information. So there were so many questions that came to mind. So remember that east-west divide? So is Lehi-Lahatsara sort of a smoking gun of east-west corridors of the past? When did their diversification commence? What will patterns of gene flow look like among and within populations? Are these relic forest museums of genetic diversity? Has there been sort of a connecting and disconnecting of these forest fragments over time so that you get these, you know, sort of gene flow, but then isolation for long periods of time, and then gene flow, reconnection, and so on, where you would build up genetic diversity in these small patches? So these are all these questions that came to mind just based on the N of 3 in context. So the sort of just really crude models that we're contrasting here is if the central plateau has been fragmented for long periods of time, it's going to be a very different sort of model of genetic diversity than continuous forest, where it's sort of a, you know, isolation by distance, you know, diffusion model. So if you're to like take a kilometer distance and, you know, collect a mouse lemur here and a kilometer away, if you're in this situation, it seems like your genetic divergence is going to be much higher than it will be in this situation because gene flow is so much smoother and readily achievable. So that's the model, that's the hypothesis. It's cut off a bit. So the traditional hypothesis of Madagascar's landscape is that it was completely forested, you know, stem to stern, east to west. And this is where you see the statistic that Madagascar is 90% deforested. And that's true, 90% of the island does not have forest. But the 90% deforestation implies that it had to have been 100% forested at one time. So that's a sort of traditional and actually French view of Madagascar. Then there's a fairly new hypothesis, the Grassland Hypothesis, that posits that endemic grasses appeared in Madagascar actually in the Miocene about 23 million years ago, and the savanna is a natural feature of the landscape, and it's quite ancient. And then there's this sort of like just right, you know, Goldilocks hypothesis that the highland plateau has actually been a mosaic for a long time, and the grasses are a natural part of that, but it's, you know, woodlands are as well, so it's sort of a patchy landscape. So we did DD-RAD. I'm going to skip right through that in the interest of time. We got lots of SNPs, so we're, you know, well beyond the 2,300 base pairs of mitochondrial DNA. This work was done by a graduate student in my lab, Ryan Campbell. I mean, we did this work so fast, it was crazy, because we had a deadline. And we did a coalescent-based divergence time analysis. I'm just going to skip right down to, so this was using BPP. We didn't have a good prior for mutation rate. So what we did was we just took a conservative approach, used the mouse and the human mutation rates and sort of used those as bounds for the MCMC model. And this work was done by Mario Disrace. So to get quickly to some results, we compared Lehi-Latsara, which is this putative plateau specialist with the fragmented landscape. We compared that with Myoxenus from the west, where forests have been continuous until the very recent. And sure enough, we see a very different pattern of genetic diversity versus linear distance. So here's geographic distance on the x-axis and genetic distance on the y. Here's Lehi-Latsara. So for a given geographic distance, genetic distance is much higher in Lehi-Latsara than it is in Myoxenus. So it seems to support our hypothesis. Looking at the time, much more recent diversification by these measures than I anticipated about 500,000 years ago for this basal radiation. This is Microcebus rufus, the real deal, here in eastern Madagascar. And here's Berthae, which is a western specialist. And this separation, by our measure, was just about 55,000 years ago. And I just want to emphasize this place is, so this is an animal that's adapted to this kind of really dry, deciduous environment way on the west coast, versus Microcebus murinus, which is found in this very wet, humid, rainforest on the east. So I think this speaks to the species issue and the rather rapid rate that speciation can occur if our dates are correct. We used also a program called SpaceMix, and just to cut to the results, what it does is it maps, you map your alleles onto the landscape, but it shows, it models what the ancestral distribution would have been of the alleles in your population. So what you see is, even though the actual localities were here in the east, there's a pull, a historical pull to the west, implying that there was some kind of connectivity between the east and the west. So that speaks to this, connectivity, and also the importance of corridors. I just want to emphasize, that part of this story, the conservation thing. So what did we conclude? That prior to human arrival, there was a mosaic of woodlands, bushland, and open areas. Fire, I don't want to minimize human impacts because they have been severe. That fire from human activities has dramatically transformed the landscape, and about 1,000 years ago, primarily causing the spread of secondary grasslands. So even though maybe there are endemic grasses, there have been secondary invasions. But that forest fragments were isolated far earlier than human arrival. And so that movement of animals from them may have occurred during times of warm cycles. This is language from another talk. Anyway, the point being that it does seem like there's good evidence, even from, starting from an N of 3, of course, in context with a larger sample, that the central highlands were fragmented for a very long time, but that there were these forest corridors that existed for some period of time. So take-home messages: we may need to revise our language, saying there's only 10% of Madagascar's forest remaining. And again, I say it again, the importance of forest corridors cannot be emphasized, because they really are mediating the gene flow among species. And when you cut those off by human agency, and this happens naturally. This is obviously what happened naturally in Madagascar, is there was the separation of the east and west probably 55,000 years ago, but we are potentially cutting off these important avenues for gene flow as we are degrading the landscape. Now, so are we right, is the question. And just like a couple of weeks ago, this paper came out where they have done time tree analysis of grasses. And their conclusion is that they recovered 69 lineages of endemic grasses, largely from Africa, 25 that were C3, 44 that are C4, and that they're the product of multiple overseas dispersals from Africa with colonization beginning in the Miocene. So they are saying in this paper that this grassland mosaic hypothesis is supported by 69 lineages, time tree analysis of endemic grasses. So this is pretty cool. Now, what are we doing about it to increase our sample size from 3? We have in Paul Hohenloh's lab as we speak, we have sent 283 individuals from over 20 species with extremely thorough geographic coverage. And we're going to be looking at lots of different questions. I know people have to go, so no worries. I've been told not to be offended, and I knew I'd be going long. So anyway, we're going to take a very close look, for one thing, at this species delimitation of these populations here that we are now no longer calling Urinus. We're going to look very carefully at this plateau hypothesis. There are, any number of projects that we're going to be working on with these data when we get them. So they're building the libraries now. They're making the libraries now, and we'll do the sequencing at Duke. So that's coming. So to conclude, the concluding set of slides is that, so we have all these questions, and I've only touched on a very couple of questions. We have, they just go on and on. So we're just building a genomic toolkit to end all genomic toolkits. And the first step has been this, what we call the Microcebus Urinus 3.0 assembly. And this just to show you, so there was the Broad Institute did the first mouse lemur genome, and this is all done with the same individual, by the way, which was very cool. So the N50 for this 1.0, 214 kilobases. Then we did some PacBio sequencing and so Illumina and PacBio. We got the N50 up to 3.7 megabases. And now with this hybrid approach, and you can read about it in this paper where we use high-C optical mapping, we just threw the book at it. We've gotten the N50 up to 93 megabases. So we're getting to the point where we basically can talk about chromosomes. And I talked about that a little while ago with the V1Rs. So, and we can look at synteny between the mouse lemur karyotype and the human karyotype, and you can see that mouse lemurs are tiny animals with tiny chromosomes, and they have a lot of them. So that's been a really interesting exercise. We also have assembled genomes for multiple species, deep coverage genomes for multiple species within the radiation. We have 15 individuals of the gray mouse lemur from the DLC and from 2 other colonies. We have 2 sets of trios from the DLC. We're looking at spontaneous germline mutation rate to see if we can fine-tune some of these divergence time estimates. And we have 8 outgroups within the lemuriform radiation that we also have good genomes for. So the bottom line is it's party time in the Yoder lab with all these genome data. And I have 3 postdocs that are there, and a graduate student and a technician. And they're just like churning through the data as fast as they can. And so stay tuned for all the fun science to come out. And so I want to end by thanking Team Mouse Lemur from 2016, Marina Blanco, Ryan Campbell, Kelsey Honeycutt, Peter Larson, Dave Swafford, and Dave Weisrock. Everybody worked really hard on that Lehilat Tsara paper, and it was great fun. And thank you very much for listening. Okay, I don't know if there's time for questions or not. I tried to get through that.
All right, that was a lovely talk. Any questions? Chuck, I see you're ready.
Yeah.
The call differences.
Chuck and I go, like, when we worked in the basement together at the Smithsonian.
Yeah.
and Stan, Stan used to provide coffee.
You've got
for
fish blood.
Yeah.
The calls differences are really dramatic. Right?
Yeah, I know. Very cool. Huh?
Are there anatomical differences in the call generating apparatus of these?
I have no idea. I have no idea. I mean, I don't know anything about mobilization. I mean, so I think the analogy would be maybe but this wouldn't, yeah. One analogy might be, different languages in humans, and we're all using the same apparatus. But of course, this would be much more deeply genetically moderated. So I don't know. The short answer is
And
I
I
don't
guess a
know.
follow-up question, do you know if different lineages respond favorably to calls
Yes.
from?
So playback experiments.
Yeah.
So Buta Ratchspiel and Elke Zimmerman have so it's very hard to have mouse lemurs outside of Madagascar, especially now. But they have a facility in Madagascar where they have 3 species of sympatric mouse lemurs, and they have done a lot of the playback calls and so on, and they show very strong preferences for their conspecifics. So yeah, they have done that, but it's not it's not like going out in your backyard with mockingbirds or whatever. So it's hard work.
Mal?
Yeah.
Sorry, her hand was raised first.
No, you go.
How many of these species are actually able to hibernate?
So the dwarf lemurs are the ones, and so they're in that same clade, the mouse and dwarf lemurs. I could talk about this for 4 hours, I swear. But so the dwarf lemurs are the true obligate hibernators. Mouse lemurs torpor, and actually Sandy Martin at University of Colorado calls them torpinators rather than hibernators, because they do this sort of facultative torporing where they can do, they do lower their body temperature to ambient, and they fall out, but it's sort of a circadian behavior, and it's very much mediated by temperature and just mood maybe. I mean, it's really hard to say, but they do. So I mean, just to go a little more deeply into it, it's a fascinating system in the mouse lemurs, because they do sometimes they will do true hibernation, which is defined as multiple days of ambient hypothermia, and so they can do that. And in some cases, there are species where the females will hibernate for 2 or 3 months. The males won't, but in a different locality, the same species, they may or may not even do daily torpor. So it is all over the map in the mouse lemurs. And with the dwarf lemurs, it's very much like genetically mediated, obligate every year, same time of year they hibernate. They go underground or in tree holes. Mouse lemurs much more flexible.
How many mouse lemurs live in one of these 1 kilometer patches? Do you have an estimate of the census population size?
Marina would be able to tell you instantly.
That's a really small fragment.
I know, yeah. So I would say maybe 30 or 50, maybe. I just don't know. But here's a. So it's really dependent on the habitat. What can be supported. So Marina, a new field site that we're going to target for looking at some of these species, looking at introgression across hybrid zones, because they do hybridize when they're sympatric. They'll hybridize, not always. But there's a site in northwestern Madagascar. It's a 1,000-hectare field site, and it's pristine. It's owned by, it's privately owned, and it has been protected for ever. Marina went there, and she said in 3 days she found 150 mouse lemurs, and that was unprecedented. It never happens. So this is where in a completely undisturbed area, you can get very high densities.
Do you have estimates of genetic diversity within one of those small patches?
Well, that's what we're working on. So when Marina is our field laborer. She has been back twice now since we collected those first, or she collected those first 3, and we now have more from Ankafu Bay. I think we have another 5 or so from Ankafu Bay, and she's also sampled 2 other forest patches. I mean, you would have noticed, in the landscape that wasn't the only forest patch, that there were others, a few kilometers away. So, and those are in the hands of Paul Hohenlohe now. So ultimately, we will have some answers for you, but we don't have them right now.
But you're not really collecting these animals. You collect them, but then you release them.
Oh, yeah,
So-
it's capture release, yeah.
Yes.
So it's ear clips
So,
and.
look, ear clips.
Yeah, ear clips, blood sometimes, and actually Mark Krasnow, who's at Stanford, who's very interested in developing, and I apologize for not also, but obviously I didn't have time to talk about, the sort of genetic model organism, and Mark is very interested in that, and his veterinarian developed a very clever technique for collecting blood where you just put a little Vaseline on the thigh and you just poke it with a pin and then squeeze, like, you know, when you poke your own finger, and then you can collect about 300 microliters of blood that way, you know, just kind of scooping it up. So blood sampling is a lot more, you know, hands-on even than your clip, just, you know, take a little clip. But it's, you know, you leave the animal without lasting marks. So Marina has been doing both, so we have blood and ear clips for a lot of these things.
So do people know if they can travel far in grasslands and other areas between these fragmented forests?
That's a great question, and Marina, again, we are outfitting the mouse lemurs in Untitled Bay with radio collars so that we can actually track to see if they're immigrating, you know, exchanging localities. And there's the movie Island of Lemurs, Madagascar. It's an IMAX movie. I don't know if you've seen it. Morgan Freeman is the narrator. It's really, I cry every time I see the trailer. I know it's ridiculous. But there's one scene where it shows a little mouse lemur leaping, you know, from one tree like to one that's quite far away, and I'm like, wow, dispersal. So I don't know. They're amazing little creatures. So I don't know, but that's a great question, you know, because that you could see it looks like a moonscape. You know, you just can't imagine that they would be able to readily get along. But you know, we're going from N of 3 to N of 20 or so in the context of a bigger phylogeographic survey. So I can't wait to find the answers, and also the radio tracking will be interesting. Yeah.
Why do they go into torpor hibernation? Is it to temperature,
Well,
or food?
I mean, that's the $1,000,000 question. It's, you know, why does any mammal hibernate? And it's, so we were talking about this quite a bit on Monday, and the answers are vague, but we came down to resources is, you know, being more important than anything else, particularly with the obligate hibernators, that, you know, if you provision an obligate hibernator, they won't actually hibernate in the true sense of the word, but they get, as we call it at the Lemur Cemetery, torpory, where they just kind of get, you know, lethargic and a little fat, and, you know, really, I mean, it's quite notable, but they don't go into the, like, I mean, and it's, you know, I think so I gave my first hibernation talk on Monday, and, you know, so I had data from Marina and others, but they go from 300 heartbeats a minute to like 3. And they breathe once every 5 minutes, and, you know, their body temperatures can go down to 9 degrees, and, you know, so it's profound, you know, the changes are profound. And then they have these interbout arousals, so, which are very expensive, you know, and, but they're, you know, so hibernation, I'm all hibernation biology right now after this workshop. So it's very expensive to do these IBAs, as they're called. You're obviously saving a lot of energy when you're down, but when you go through an IBA, you're, you know, revving all the engines full, you know, and like that. You have to turn every system on, you know, fast, and it's going full blast, and then you come back down. And so the energetics, you know, it's a sort of a wash almost. So it's interesting.
I was just going to say, there are a lot of, you know, small mammals that undergo daily torpor cycles for energy and conservation purposes, and if you deprive them of food, you can extend that daily torpor into
longer periods
of time. So, I mean, that's been shown experimentally.
Right,
so
sure.
it could be food, you know, generated. It could be temperature generated. There's a number of factors that can, for facultative to prolong hibernation period.
Yeah.
Is the phenology of resources periodic in the way that they're torpors?
Yeah, so it coincides with the dry season in Madagascar, but as we, you know, discussed, so that you can have hibernators living symptotically with other mammals, some closely related perhaps, that aren't hibernating. So why choose hibernation? And, you know, and this is where I like to go back to Darwin and natural selection. You know, it's not perfecting, it's the good enough solution, right? So it's a strategy that works, and it got fixed somewhere, and yeah.
Don't they become incredibly vulnerable to predation?
That's a great question.
Yeah.
That is like that digging up the dwarf lemur underground. So Fusa they're the real predators in Madagascar, and they're very good at sniffing out lemurs. They'll walk right over the ground so there's, so all cellular function is basically shut down, mitosis, everything, when you're in the full, hibernating state. So any chemo signals that they might be producing probably, or I don't know, nobody's looked at it yet. And there was a woman at this conference who said, oh, well, she just had all kinds of understanding of the biochemistry of the skin. And she was saying, you can figure this out very easily by taking a skin biopsy of a hibernating dwarf lemur and, look for X, Y, and Z, and then you will know. And I'm like,